Hello, my dear student. I am Krishna Kumar. Today we discussed about the reproduction in human. As first we start here, development of embryo. Development of embryo takes place in the female reproductive system through following steps. Every month a single egg comes out of the ovary and reaches the fallopian tube of female reproductive system. During copulation, sperm reaches the fallopian tube where a sperm fertilizes the egg. This results in the formation of zygote. When the zygote is developed, the zygote divides repeatedly to give rise to a ball of cells. The cells then begin to form groups that develop into different tissues and organs of the body. This developing structure is termed as embryo. As we see in these structures, the balls of cells, this is the embryo that develop from the zygote. Here is the number of cells is get multiplied, developed and form a ball-like structures called embryo. The embryo gets implanted in the wall of the uterus for further gradually developed the body parts such as hands, legs, heads, eyes, ears. And the stage of the embryo in which all the body parts can be identified is called fetus. And the process by which the embryo is get attached with the wall of uterus is known as implantation. When the fetus is developed, then after completion of the 9 month, the mother gives birth to the baby. As we see in this figure, the fetus is into the mother's womb in uterus. The uterus is completely developed by their body organs, and now when the 9th month is completed, the maturity age is completed of the baby, then with the labor pain, the mothers give birth of the baby and the new baby is get born. Now come to the formation of egg cell in hens. Internal fertilization takes place in hens. After fertilization, the zygote divides continuously and moves to the oviduct. As it travels down, many protective layers are formed around it. The hard cell in a hen's egg is one such protective layer. After the hard cell is formed, the hen finally lays the egg. The embryo takes about 3 weeks to develop into a chick. The hen sits on the egg to provide sufficient warmth. After the chick is completely developed, it bursts open the egg cell and the newly born chicks is come out from the hens after hatching the egg. Now we say about the viviparous and oviparous animals. The animals which give birth to young ones are called viviparous. For example, human beings, dog, cow. Those animals which lay eggs are called oviparous animals. Example, hen, frog, etc. Now we discuss about the IVF, in vitro fertilizations. A biological process carried out in laboratory is called in vitro. Thus, fertilization carried out in laboratory is called in vitro fertilization. As we know, some women are unable to bear babies because of oviducts are blocked. In result, sperms cannot reach the egg for fertilization. Doctors collect freshly released egg and sperms and keep them together for a few hours for IVF or in vitro fertilizations. If fertilization occurs, the zygote is allowed to develop for about a week and then it is transferred in the mother's uterus. Whole development takes place in the uterus and the baby is born like any other baby. As normal baby, the baby is developed. Babies born through this technique are called test tube babies. Young ones to adult. Direct development of the baby maybe takes place when the young ones of an animal resemble the adult. That direct development takes place as for example in hen, man, monkey. You can see here in hen how the direct development of the baby is takes place by the laying of the eggs. Indirect development, when the young ones of an animal do not resemble the adult, then indirect development takes place as like frog, butterfly, silk moth, in which their life cycle, the egg is produced, but after the development from the egg to adult, there is the drastic changes can be seen and they are not resembles to the adult. So such type of indirect development is takes place in the most of the organisms as like frog, butterfly, silk moth, and these are known as metamorphosis. In case of indirect development, transformation of young ones into adult through drastic changes is called metamorphosis. Larva of butterfly undergoes metamorphosis to become a butterfly. A tadpole undergoes metamorphosis to become a frog. Now we see the asexual reproduction. In this method, the process of reproduction in which new individuals are produced from single parent. In microorganisms, the asexual mode of reproduction takes place. We discuss here the two mode of asexual reproduction, budding and the body fission. The budding, this reproduction methods is seen in those multicellular animals which are highly simple in structure. A small bud or bulge develops on the body. After developing the bud, it gets detached from the parent's body to begin life as a new individual. Since new individuals develop from the buds, this type of asexual reproduction is called budding. For example, hydra and sponge. Binary fission. This method of reproduction is seen in unicellular animals, example amoeba. In this method, an organism divides and forms two daughter cells. First, the nucleus divides and forms two daughter nuclei. Then the cytoplasm in the mother cell divides into two daughter cells. This leads to the formation of the daughter cells, each having a nucleus and its own cell organisms which then develop into a fully formed adult. For example, in paramecium in amoeba, the binary fission takes place. As we see in this figure, the parental body is get divided into the two daughter cell by dividing their nucleus into two, and cytoplasmic division is also takes place. Now we discuss about the cloning. Cloning is process of production of an exact copy of a cell, any other living parts or a complete organism. Cloning process of an animal was successfully performed for the first time by Ian Wilmot and his colleagues at the Roslin Institute of Edinburgh, Scotland. They cloned successfully a sheep named Dolly. During the process of cloning, sheep named Dolly, a cell was collected from the mammary gland of a female fin dorsate sheep. Simultaneously, an egg was collected from a Scottish blackface ewee. The nucleus was removed from the egg. After that, the nucleus of the mammary gland cell from the fin dorsate sheep was inserted into the egg of the Scottish blackface ewee, whose nucleus had been removed. Then this egg was implanted into the Scottish blackface ewee. Development of this egg followed normally and then finally Dolly was born. So the Dolly is the first baby of the sheep that is cloned Thank you, my dear students, for watching my video. Again, I come with a new video in the next class. Thank you.